That's what Brad is.
I like
So we have exam next week, I believe, next Wednesday. It will cover chapters 5, 6, and 7, which is certainly more than enough in my opinion. There's a lot of stuff here. And so we need to do chapter 7 this week, and then next week, Monday, of course, will be another practice exam. And then Wednesday we'll do the real exam. So chapter 7 deals with photosynthesis, which you'll find in many ways is the exact opposite of glycolysis, because in glycolysis we took glucose molecules, broke them down to 3-carbon compounds, and then extracted high-energy electrons out of those to produce ATP. What plants do and other photosynthetic organisms is, in a sense, the opposite, because they take carbon dioxide, which is a waste product of glycolysis and Krebs cycle and electron transport. They take CO2 and they use it to make 3-carbon molecules, which are then used to make things like glucose. So they go in the opposite direction. The energy source, of course, is light energy. All right, so major points. Photosynthesis is, we might call it the backbone of the biosphere. The biosphere being the entire area of the Earth that supports living organisms, which is darn near all of the surface and down in soil at least a meter or two, up into the atmosphere quite a bit. But that region is supported completely by photosynthesis, because that's where the energy to produce organic molecules is captured. Right? It's no mistake that we call photosynthetic organisms producers, because they really are producing energy molecules for all other organisms to use. Okay, so nearly all ecosystems depend on photosynthesis. The only ones I can think of that don't are systems in caves, for example, where light doesn't penetrate, or in deep sea vents. And interestingly, there is light at deep sea vents. So there are potentially photosynthetic processes that could be occurring down there as well, but much slower. But they depend much more on other chemical energy sources. Photosynthesis happens in 2 steps. Okay, the light reactions and the Calvin cycle. You've probably heard these as the light reactions, the dark reactions. I don't like the term dark reactions because people think they only happen in the dark. They don't. They happen all the time. They're just independent of light. Okay? The first step is the step in which chemical light energy is captured to produce chemical energy, ATP and high energy electrons. And then the second step, the Calvin cycle, is used to actually make the 3 carbon molecules that will be used to make sugars and other organic molecules. Both prokaryotes and eukaryotes do photosynthesis. There are photosynthetic prokaryotes. There are photosynthetic eukaryotes, plants being the obvious example, but there are plant-like protists that do photosynthesis as well. The methods differ a little bit between them. Some prokaryotes use completely different molecules to do photosynthesis than do eukaryotes. So we'll talk a little bit about that. All of these organisms are autotrophs. I don't know if I've used that term before, but that means that they make their own organic molecules. That's a hallmark of photosynthesis. That's what's happening in the combination of the light reactions and the Calvin cycle. But they still require a carbon source, a nitrogen source, and some minerals to grow. So you have to have those things available. The source of carbon is typically carbon dioxide. Right? So each of these photographs illustrates a different facet or ecosystem in which photosynthesis is occurring. And so I thought I'd just go through each of these. These are the typical land producers. I mean, there's a nice oak tree, some flowering plants around it, some herbs. And those are all producers. As I said, producers are the organisms that are actually capturing the light energy, producing organic molecules, which serve as energy sources for essentially every other organism that depends on them. Okay? So there's the definition of a producer. Without these producers, ecosystems would collapse, at least the ones that we know about. As I said, deep sea vents, cave ecosystems, they're not as dependent on those producers, but every other ecosystem is. So without these kinds of organisms, every ecosystem would fall apart. These are also using the typical photosynthesis processes that I'll talk about. There are some variations of photosynthesis that plants do. Some plants do things a little bit differently than other plants. And so your book goes through that, and so I'll go through that as well. For example, these are cactus. Cactus are also producers, but they're in the New World only. Most people don't realize that cactus don't exist in the Old World naturally. So you don't find them in Europe, you don't find them in Asia, you don't find them in Africa naturally. Okay? They are only North and South American organisms. They live in very arid environments typically. However, there are some cactus that live in rainforests, so don't jump all over me for saying they're arid plants, because there are some that live in very wet locations. But they all use a modified photosynthesis that's designed to be able to be used in a very dry environment, and it's called CAM photosynthesis, or crassulacean acid metabolism. And I'll talk about that process towards the end of this slideshow. Ocean plants are incredibly important not only for ocean ecosystems but for land ecosystems as well. The plankton, phytoplankton, or plant plankton, single-celled protists that live in the open ocean are the most important oxygen source for our world. They produce far more oxygen than any other ecosystem just because there's so darn many of them. So they serve as oxygen source, they serve as a primary food source for aquatic organisms. Okay, so ocean ecosystems could not do without those organisms. These larger algae like kelp, this is a kelp forest, kelp are critically important in aquatic ecosystems as well. Right, and finally, this, what looks like a brown sludge is actually purplish, and it's full of purple bacteria. They use a molecule called bacteriorhodopsin, which gives them their purple color, but that's the molecule that's being used for photosynthesis. Much different than what plants do. Okay? Plants don't use bacteriorhodopsin. We actually do use a form of bacteriorhodopsin in our eyes. Right? It's a molecule that allows us to see black and white, grays, and colors. All right, so we have a variation of these molecules that we're using for sight because they're light sensitive. Okay, here is an overview of photosynthesis, much like the overviews I gave of glycolysis. And as you look at this, you'll see that it's almost glycolysis in reverse. You have carbon dioxide and water, which are the waste products or breakdown products of glycolysis. And with light energy, you're going back to glucose and oxygen, which are the inputs for the cellular respiration processes. So this really does look like that process in reverse. However, the actual details of the process are very different. Okay? So glucose is assembled rather than broken down. It's assembled in steps, though. We'll find that in the light reactions, you don't make glucose right off the bat. You make 3 carbon molecules, which are then used to assemble a number of other molecules, including sugars. All right? And the process uses carbon dioxide and water to do this. All right? So one of the reasons these are called producers is because, of course, we consume that glucose, other organisms consume that glucose, because it's a good energy source. And so we can break down the energy that's been captured by those producers from light. And that energy is streaming down on us all the time. Photosynthesis occurs in the leaves of plants, some in the stems, but mostly in the leaves. Of course, cactus, lacking leaves, do it all in their stems. The stems have been modified to become photosynthetic structures. And there are some other funny plants that actually don't do much photosynthesis at all. They depend on other organisms for their nutrient sources. Okay, so this figure I actually took out of chapter 31, but it shows the leaf structure in cross-section, and I think it's important to understand where this is happening so you can keep a perspective on what's going on. There's basically 4 tissue layers in this leaf. The first layer is the upper epidermis. Notice how blocky those cells are, and notice how clear they are. All they do is protect the surface of the sublayers of the leaf from water loss and damage. Okay, so you have these square cells. They're very clear. They allow light to flow completely through. The next layer down is where most of the photosynthesis in the plant, leafy plant, occurs. That's called the palisade layer, or the palisade mesophyll. Mesophyll meaning middle plant, referring to one of the middle layers of the leaf. Notice these cells are all column-shaped. There's usually 2 layers of them, but looks like they're only showing 1 here. And there's lots and lots of chloroplasts in these cells. They're packed very, very tightly together. And so that way they can maximize the absorption of light that's streaming down from the top of the leaf. All right, so that upper epidermis is actually facing the sun. Below that is a more open layer called the spongy mesophyll, or spongy layer. As you see, these cells are much more irregularly shaped. There's lots of open spaces between them. Right? There's a reason for that, because that those open spaces allow for gas exchange to occur. They allow for carbon dioxide to come in. They allow oxygen to get out of the leaf tissues. Okay? And then finally, the bottom layer is the lower epidermis. That's the layer that's shaded from the sun, because the sun is sort of beating down the upper epidermis. There is some photosynthesis that happens in this spongy mesophyll, but not a whole lot. Certainly not as much as the palisade, because by the time the light gets down to the spongy layer, the palisade has absorbed most of it. So not a whole lot gets down there. Okay, the other thing you see in here is openings in the leaf. Those openings are critically important because they let carbon dioxide in. CO2 is not going to diffuse very easily across the cell walls of the plant. And so in order to get carbon dioxide up through the spongy layer and up to the palisade where photosynthesis is occurring, you've got to have those openings and gaps in the spongy layer, and you've got to have openings in the epidermis itself. All right? That's provided by these guard cells which form an opening called a stoma. Plural is stomata. Okay? So the stomata can open up, allow gas exchange to occur. When it gets dry, what happens is those cells lose turgor pressure, they start losing water, and so they close up, and the stoma closes. That's important because that prevents water loss. It prevents dehydration. But it causes a problem because it traps oxygen. And as we'll see down the road, having oxygen around when photosynthesis is trying to happen is a very bad thing. All right, so some plants have found funny, interesting ways of getting around this. The other thing you see in this leaf is some vascular tissue. It's actually been circled up there. That's conducting tissue that conducts sugars that have been produced down from the leaf and water from the roots up to the leaves. Okay, it's called xylem and phloem. Don't need to worry about that now. Okay, so we have our leaf. We look at the cross section, and there's our two mesophyll layers here and here. The palisade is right here, the spongy layer here. Here are the stoma or stomata that are opening up to allow carbon dioxide in and oxygen out. If we look at a single cell, in this case they've picked one in the spongy layer, here's a spongy mesophyll cell which is just jam-packed full of chloroplasts. Okay. You can sort of see the central vacuole in between these chloroplasts. These chloroplasts are actually squashed up against the edge of the cell, and you're looking at it sort of front on. Okay. So these are all squashed up against the cell wall, and the vacuole is inside. All right, we zoom in on one of those chloroplasts, and here's the same figure basically that you saw in Chapter 4. Typical chloroplast, it's got three distinct compartments: the intermembrane space, the stroma, and the thylakoid space. Okay. The actual membranes around the chloroplast don't have light-capturing pigments in them. It's these thylakoids, right, that have all those photosynthetic pigments in them. And so that's where photosynthesis is going to be occurring in the plant. And then again, just for comparison, here's an electron micrograph of the chloroplast showing those stacks of thylakoids. All right, so it's the thylakoid compartment in the stroma that's going to be important in terms of understanding how photosynthesis works in these plants. All right, so oxygen is produced by photosynthesis. You can demonstrate that without really knowing anything about the process because that gas that's put off by the plants supports a flame. All right, so you know it's oxygen. This is a water plant. Elodea is the genus. The leaves are only 2 cell layers thick, and so they're able to capture quite a bit of light. They look sort of transparent. But they do a hefty amount of photosynthesis, and that's being illustrated by these oxygen bubbles that are forming on the leaves. Now, oxygen doesn't diffuse into the water very well. Oxygen doesn't hold water doesn't hold oxygen as well as air does. And so this stuff doesn't dissolve, and so you get these bubbles of oxygen coming off the plants. All right, so how is that oxygen produced? What molecules contribute to the oxygen that's produced in photosynthesis? And this question has been investigated in a number of ways. We know that it comes from water. And so water is somehow being broken down. That produces oxygen, which is released by the leaves and is allowed to leave the photosynthetic tissues. Okay? Water serves as a donator of electrons. That's how it's going to function. What's going to happen is sort of the opposite of what happens in cellular respiration. In the mitochondrion, what happens is electrons are donated to water, which splits it, or donated to oxygen, which splits it to form water. Okay? In photosynthesis, the opposite happens. Water is split to provide electrons, and as a result, you produce oxygen. And so these are classic redox processes where electrons are being snatched away from molecules. Right? The way that you understand or try to investigate this is by using radioactive isotopes. And these are the experiments that were done. Right? In this first experiment, radioactive carbon dioxide was used. Right? So there's our labeled oxygen. We can follow that through photosynthesis and ask, where does the radioactive oxygen end up in this process? Okay. It turns out it ends up in the glucose that's produced. It ends up in some water that comes out of the process. And it does not end up in the oxygen that's produced. So we know that the oxygen that's coming out of photosynthesis is not coming from the carbon dioxide that's going in. Instead, if you take water and label that oxygen in water by using, say, a radioactive isotope, follow it through photosynthesis, you find that the oxygen does, in fact, get labeled. Right? It is radioactive. And so you're able to make the conclusion that the oxygen is coming from the water. Right? Now, why is this so important? Because in the 1950s, when these experiments were being done, nobody had all those molecular tricks and tools that we have nowadays. Right? So this was a very good tool for charting molecules going through biological processes and asking how do those molecules get rearranged, where do the atoms go, how do they get pulled apart and pushed together to produce the products. Right? It gives you an understanding of what's going on without having to have all of the molecular details. Okay? So in the first experiment, labeled oxygens end up in glucose and in water, and in the second, only the oxygen gas produced was labeled. So we know that the water going in is being busted to make oxygen coming out. In fact, using this kind of logic, you can label any of the molecules that are going into photosynthesis and ask, where do those molecules come out? We find that the carbons are coming out in the glucose. No surprise there. Okay, so those carbon dioxides are being stuck together somehow to produce glucose. The oxygens from the carbon dioxide are ending up in glucose and in some water that's coming out of the process. The hydrogens are coming from the water back into the glucose, and then the oxygen from the water is coming out, or the oxygen atoms in the water is coming out in oxygen molecules. Right, so you can track all of these molecules through, and it's really sort of amazing that this was done in the 1950s, and nobody had all of the nice little tools that make our lives so much easier in science now. Okay. So there was a pretty good understanding of what was going on, at least conceptually, in photosynthesis, but the details have taken a long time to work out. All right, so what can we conclude from these experiments? Photosynthesis is a redox process. Okay? This figure shows where the reduction and oxidation is occurring. We're reducing these carbon dioxides to produce glucose. We're oxidizing the water, stripping electrons away to produce oxygen gas in the process. Okay? And if you just compare this with cellular respiration, it's, again, almost exactly the opposite. In cellular respiration, we just sort of flip this reaction and flip the redox products. The glucose is being oxidized to produce carbon dioxide. We're stripping electrons away. The oxygen is being reduced. Electrons are dropped off to it to produce water. Right? So in a conceptual sense, at least, photosynthesis and cellular respiration are opposite, exactly opposite processes. And that may help you to remember these reactions, at least in a conceptual sense. Doesn't help you much with the details. Okay, any questions up to this point? All right. Okay, we're about ready to go into some of the details of this process. As I said, there are two stages of photosynthesis. The light reactions, which do in fact require light energy to proceed. This is where the water is split to make oxygen. Okay, so oxygen gas is coming off during the light reactions. You won't detect oxygen gas being produced if you keep leaves, for example, in the dark, but you will detect glucose being produced. Light energy is trapped. So you're taking electromagnetic energy, trapping it, converting it to chemical energy in the form of ATP and NADPH. Now, as I said, I think last week, I mentioned that NADPH is identical to NADH. So it fulfills the same function. It holds two high-energy electrons. The only difference is it has a phosphate group on it. Why do plants use the phosphate group? I have no idea, but they do. Okay, so when I say NADPH, just conceptually think NADH, and you're okay. It requires light. Right? You will not get this reaction going on unless there is light. And not just any light. Certain wavelengths of light are better for this reaction than others. The Calvin cycle, then, is the second step. The dark reactions, you've probably heard them called. In this process, you have ATP and NADPH energy being used to produce glucose and other organic molecules. Okay, so carbon dioxide is attached to existing carbon chains that the plant has. And that's called carbon fixation. As I said, it's independent of light. It occurs whether light is present or not. As long as the ATP and NADPH are available, this reaction will occur. All right, here is a summary of these two reactions without going into a lot of details. Our light reactions, that's the first step. Light energy is absorbed. There's some funny stuff that goes on here. Water is broken down to produce oxygen, and we end up getting high-energy electrons and ATP. That ATP and NADPH then is used in the second part of the photosynthetic cycle, the Calvin cycle, and that takes carbon dioxide, binds it into organic molecules, and our outcome is sugars, but not only sugars like glucose, but many other organic molecules too. Our direct outcome actually is a 3-carbon molecule, glyceraldehyde 3-phosphate. And in fact, you saw that in glycolysis. That's one of the breakdown products when you split that glucose molecule. Okay, so the light reactions that happen in the grana, those stacks of thylakoids, and then in the stroma, that's where the Calvin cycle is happening. All right, so before we go into the details, we're going to sort of take a sideline here and look at what electromagnetic and light energy is. If you want this in any kind of more detail, take physics, because I'm not going to give you a huge amount of detail on this. The idea is that light energy behaves as wave energy. That's been well demonstrated. Any electromagnetic energy behaves as waves. Right? You have a distinct wavelength. And you can imagine the wavelength as sort of the ripples on a pond. If you drop a stone into water, then you get these ripples coming off. That's the energy that's being released from the dropping the pebble in. Okay? So light energy has wavelengths as well, and that's the distance between the crests of the waves. Just measure that distance, and that's the wavelength of the electromagnetic energy. The electromagnetic spectrum includes lots of different types of energy, not just light energy. All right, light also behaves as particles. It's sort of this dual nature of light. It behaves as waves, but it behaves as particles as well. It's almost as if each packet of light is a particle with a certain energy associated with it. Right? And those packets of energy are called photons. Right? So every type of electromagnetic energy has photons associated with it. Okay? And then photons of smaller wavelength are more energetic than photons of high wavelengths. And you can sort of graph this out on an electromagnetic spectrum. Here. Right, this is a simplified electromagnetic spectrum, and all we're doing is looking at the wavelength of light. There's a very narrow region in here that not only is used by plants for photosynthesis, but is also used by us for perceiving light energy. This is visible light, and it goes from about 380 nanometer wavelength, which is the deepest purple that we can see, up to about 750 or so, which is the deepest red that we can see. Beyond this, on the right-hand side, we get into infrared. Those are higher wavelengths. And then lower than 380, we get into ultraviolet radiation. So you can see on this chart, ultraviolet, X-rays, gamma rays. Gamma rays have exceptionally small wavelengths, and they're extremely energetic. You don't want to be exposed to gamma rays. Right? Even X-rays cause some damage. All right, so everything down in this side of the table is very, very energetic. Up on the right side of this, these are less energetic, and it's a darn good thing because we're getting bombarded by radio waves, while we're sitting here. It would kind of suck if those were damaging, but they aren't. Right? Why is it that there's this very narrow set of wavelengths that we use for perceiving light and that plants use for generating photosynthesis? Right? The reason is because they're just the right energy. They're able to excite molecules in the ways that are necessary to absorb that energy, but they're not so energetic that they're going to damage those molecules. Right? Things above about 750 nanometers are not energetic enough to excite those molecules, so we can't use them. Okay? So UV and lower is too energetic. It will damage organic molecules. Okay? We know very well that UV, X-rays, and gamma rays will all damage DNA. All three of those can break DNA molecules. They can break chromosomes. They can cause other kinds of damage in DNA as well. And they'll damage other molecules, so we can't use those. Infrared and higher are not energetic enough, okay, in the right ways to be able to use those to perceive light or to absorb light energy. And so it's no surprise that plants use the same wavelengths that we do for light energy. Yes?
They're low enough to not cause damage, but as we have more and more of them in the air,
Well, yeah, I mean, as you have more infrared radiation, obviously you feel that as an increase in heat. Sooner or later, you're going to get to the point where that might be damaging. But no, if you're exposed to a given wavelength of, say, infrared radiation, it's not going to hurt you. Yeah, same thing with radio waves. So all of you who have avoided using cell phones because there's been some talk that the cell phones produce this electromagnetic radiation that causes brain cancer, doesn't. There was actually a case that recently got thrown out of court on that. There's just not enough evidence to even prosecute it.
is there some radio waves that go through there, or some kind of?
There may be some electromagnetic radiation because there's a magnet in there, so you're getting magnetic radiation and electromagnetism are tied together, but it's not enough apparently to cause any harm. Right? That's the same way with this magnet therapy that some people use. It's been shown that those magnets that you buy at like the Bon or whatever, the magnetism doesn't even penetrate the skin. It can't even penetrate frog skin, which is thinner than ours. Right? So it apparently doesn't do any good. It's all psychosomatic, which that's okay. I mean, if it makes you feel better, great, use it. But it's not the magnets that are doing it, it's your brain power that's doing it. All right, so the result of this absorption of light is that most of the wavelengths in plants are going to be absorbed. Right? Most of the wavelengths that strike plants will be absorbed. There's only a few colors that aren't. Okay, green, for example, that's the obvious one that gets reflected off. There's a few rare additional colors. If you see some purples in plants, that means those purples are being reflected off as well. Right? So we perceive those colors because they're being reflected off. So plants look green, or mostly green if you're a purist. All right. Photosynthesis uses a number of pigments. You've all heard of chlorophyll, sort of the classic photosynthetic pigment that's used in these light reaction processes. But there are a number of other pigments that are used. Chlorophylls can only absorb specific wavelengths, and so they're not useful for much of that electromagnetic or visible light spectrum. You've got to have something else to absorb those wavelengths, and so plants have a whole bunch of molecules. Chlorophyll A and B, which absorb blue, violet, and red most efficiently. So that's fine if all that's bombarding down on you is blue, violet light, and red light. But white light has all of those frequencies in it, and so you need other pigments. Carotenoids, which absorb oranges and yellows best. Okay. Phycocyanin, which absorbs oranges best. Phycoerythrin, which absorbs yellow-green and yellow best. So if you have all of these pigments, what that means is that you've got all of those wavelengths covered, except for green, which plants don't really have any pigments to absorb. Right? So almost any of those visible spectrum photons can be absorbed by plants by one or another of these pigments and used to power photosynthesis. Right? The problem is, how can you use all of these? How do you tie all of these together so that you can absorb that light energy and capture it? Right? So we need to look at that. If you absorb a photon, right, it means that you've basically absorbed the energy inherent in that photon. And typically what happens is electrons get popped up to higher energy levels in the molecule because of that, or in a specific part of the molecule. Chlorophyll, for example, has a ring structure with a magnesium ion right in the middle of it. That's what absorbs that light energy. So the electrons, the magnesium electrons, get popped up to a higher energy. Look at magnesium. It's element number 12. So it's got 12 electrons, and that means the first shell is filled with 2, second shell is filled with 8. The last shell has 2 lone electrons. If you pop those 2 electrons up to a higher energy level, it's much easier to snatch them away. That's typically what happens when photons are absorbed. Electrons are popped up to higher energy. Okay? So they go up to a higher orbital. Most of the time they pop back down almost immediately. This is what fluorescence is. It's just absorption of light at one wavelength and emitting that light at another wavelength, usually a longer wavelength. So for example, a molecule may absorb purple light and emit blue light because the blue is a longer wavelength. Or it may absorb blue and emit green. Some of that energy is lost. We know that because energy reactions are not 100% efficient. Some of the energy in the absorption and release of that light, of that photon, is lost as heat. So you have to have a lower wavelength. All right, but if those electrons can be captured, if you've got a molecule that when those electrons pop up to that higher orbital, that molecule can snatch them away and capture them, like in ADPH, then you can store that energy. So you have high energy electrons. So here's an example. If you just isolate chlorophyll and throw light at it, what's happening is that light is being absorbed, electrons are being popped up to higher energy levels in that magnesium atom, but then they pop back down almost immediately and they release energy. They release a photon of a longer wavelength and they release some heat. Okay? You increase the entropy. So that's what's being shown in this photo on the right side of this figure. Light is being shown onto this chlorophyll mix, and the chlorophyll mix is radiating light at a longer wavelength, in this case sort of a red wavelength. Okay? But it's also heating up.
So the light is the blue stuff that we're seeing?
Yeah, the light is actually the blue stuff that you're seeing sort of reflecting off the glass there. Right. All right. However, if you could capture that energy, okay, you could capture those electrons in an electron acceptor, you'd be much better off because then you can pass those electrons off to other molecules or down an electron chain, just like the mitochondrion does. And as a result, you could produce ATP. Right? So the energy can be captured and used to power other cellular processes, like an electron transport chain. In fact, this is exactly what plants are going to be doing. Right? Light is going to be absorbed by chlorophyll and other pigments. Electrons are popped up to the higher energy. They're captured, passed down an electron transport chain, just like the mitochondrion, and that's used to pump hydrogen ions across a membrane, which produces ATP. Okay. But how are all the wavelengths being used? What we need to do is we need to look at how these things are organized in the plant. Okay, these pigments are not just scattered around in the plant. They're organized into clusters of 200 to 300 molecules. And you don't just have one type of molecule in that cluster. You have many types of molecules. You may have phycoerythrins, carotenoids, chlorophylls, all arranged into those complexes or clusters of pigments. Right, the clusters are called antenna complexes, and that's where that light energy is being absorbed. They're associated really closely with the proteins that are doing the electron transporting through the membrane. Okay? So they actually are a physical part of that. So the electrons can be passed off very quickly to these molecules. This is what they look like diagrammatically. We've got photons coming in. Those photons then hit these molecules on the outside of this antenna complex. What happens then is the electrons in these molecules are popped up to higher energy, but they pop back down because there's nothing there to accept the electrons. Right? So they jump up, jump back down, they fluoresce. They release that light at a little bit longer wavelength. So then that light pops to another one of these molecules. Electrons pop up to higher energy, pop back down, release the light at a longer wavelength. Then to the next one, and the next one, and the next one, until you finally bounce that light to the reaction center. That just happens to be the molecule that is able to pop electrons up to higher energy and have them snatched away. It's the one that's associated with the proteins in the electron transport chain. Okay, so you just pop those photons from one molecule to the next, losing energy every time until it hits the reaction center. And nothing special about that reaction center, just happens to be the one that's closely associated with the proteins of the electron transport chain. Right? In plants, we find two different photosystems. The P700, or photosystem 1, and the P680, or photosystem 2. These are named based on the wavelength of light that the reaction center absorbs the best. So the P700, or P1, absorbs 700 nanometer light best, red light. Okay. P680 absorbs a little bit lower wavelength. 680 is still sort of in the dark orange to red range. Okay, but the light that came into this reaction, or this antenna complex, might have been 350 nanometer light. Pops in, loses a little bit of energy, goes to longer and longer and longer wavelengths until it hits the reaction center. At that point, you've got absorption. All right, we find then that there are two ways for plants to make ATP using these processes of photosynthesis. Cyclic electron flow is just a system where you absorb light, you pop electrons up to higher energy, you pass them through an electron transport chain, and then you just pass them right back to that photosystem again. All right, so they just cycle through that process. It's real good for making ATP. You don't make NADPH this way, but in bacteria, a lot of times they don't need to make NADPH. Okay, so the electrons are just returned to the photosystem by this process. And it generates ATP, but not NADPH. The non-cyclic flow is the one we're sort of more familiar with. That's what most plants and photosynthetic protists do. Prokaryotes do things a little bit differently. But both photosystems are involved, photosystem 1 and photosystem 2, that generates ATP and NADPH, and that can be used in the Calvin cycle to make sugars. All right, so in cyclic electron flow, we have the photons being absorbed by the antenna complex, bounced from one molecule to the next, increasing in wavelength each step until they're absorbed by the reaction center. This is typically done by a P1 photosystem, a P700 photosystem. Those electrons that are popped up are accepted by an electron acceptor. They're snatched away, and then they just pass through an electron transport chain until they're passed back to this molecule in the reaction center. Okay. As a result of passing those electrons through, you can pump hydrogen ions through a membrane. Okay. In this case, it's the thylakoid membrane. And that allows you to make ATP. Right? You get lots of ATP by this process. And it's sort of a regenerative process. You don't have to break down molecules in order to do it. You just absorb light energy. It's a really efficient process. Right? So the electrons come back to the photosystem. In non-cyclic electron flow, however, right, we have a lot more stuff going on. We have photons being absorbed by two different photosystems. We have electrons coming in from several different locations, from water, from photosystem 2. We have two electron transport chains that are doing different things. It's a much more complicated process in these plants that do this kind of photosynthesis as opposed to the simple cyclic photosynthetic process. Okay. I don't want to get into the details of this right now, so we'll go ahead and stop there and start up on this next time.